Good morning. I'm Jack Berryman, the ACSM historian, and we're here this morning interviewing Claude Bouchard as part of the ACSM's Distinguished Leaders in Sports Medicine and Exercise Science. So thank you, Claude, for taking time to come and talk, and we appreciate it very much.
Thanks for having me, Jack.
What I wanted to start with was just a little bit about your early life, really. High school, college. What did you major in? Where your first job, that type of thing. Then we'll talk a little bit about where your career intersected with ACSM, and then hopefully have time to talk about a lot of your current work, because you keep appearing in the news. And we'll talk about that too, your recent New York Times thing about the heart. So anyhow, where did you go to high school and college? And
Okay, well, you need first to remember that I come from Quebec.
Okay.
Quebec City, Canada. And the system was not quite, and still is not quite like here. So I went to, the equivalent of high school was what we call the classical college, where we study languages like Greek and Latin, and philosophy and the humanities. I did a lot of that. And then from there, I went to Laval University, where I majored in physical education. After I got my degree, and now we're going back to 1962.
I was going to ask you, when are we talking about here? In the early 60s?
That's right, 1962. After looking at many options, I decided to do a master's degree at the University of Oregon in Eugene. So I spent about 15 months there, did my master's degree. And then I wanted to do my Ph.D., but I did not know exactly what I should focus on. I had not yet made up my mind, had too many options. So I said, I need to take more experience in research. And through the grapevine, I had heard a lot about the Hdl Cholesterol in Cologne, Germany, where they had a lot of research in sports medicine, cardiovascular physiology, etc. So I went there and spent almost 2 years there.
Wow.
At the Heart Institute, embedded into the Deutsche Sporthochschule.
And this was just with your master's degree?
Yes.
What did you do your master's research in?
But I was not allowed to do a research project. It was only courses, because I was a foreign student, and my English at that time was very rudimentary.
Okay.
Not any better today, but they did not want me to write a thesis, because it would have been too much work for them.
So how about your German then?
I learned German there on site, and became pretty proficient in German.
Okay.
And I published a lot of papers in German, actually. All my early career papers are in German.
Wow.
Maybe 15 to 20 of them. And then after that, I got job offers. I didn't have my doctorate, but I got job offers because the field of exercise, physical education was very embryonic in Canada, and in the province of Quebec in particular. So I decided to go back to Laval University, where I did my undergrad work. I had an agreement with them. You stay with us about 3 years, and then we will sustain you while you do your Ph.D. Financially sustain. That was a great option.
That's perfect, yeah.
But I got caught into the whole exercise, and I stayed there 9 years instead of 3.
So mid-60s to the early 70s, or what time?
Yep, until 74.
Okay.
And at that time, I had made up my mind that I wanted to do genetics. I had discovered genetics, and I said, this is a very promising area of research. I need to learn what it is about and become competent in this field to develop a new research line.
In 1974, you were, that's early on, really, when you think of, I mean, that's...
Yeah, there were not many papers in that area.
Right, right.
So I decided to go to the University of Texas at Austin. And there I worked with Bob Molina and with people in zoology, in psychology, in cellular biology. I developed a personal track and curriculum to fit my needs, and I really had a grand time.
Yeah, that's a great campus, and I know Bob Molina very well. There's neat people there. Yeah.
So I got my degree in population genetics.
What year did you get your PhD?
77.
Okay.
And then went back to Laval, where I established my research unit in this area, and stayed there until 1999. I trained a lot of students, and after maybe 6-7 years began to have postdocs, and I got about 35 postdocs over a period of 15 years.
Wow. So you were offering a PhD at that time?
Yes.
Yeah.
Yes. When I came back, I petitioned and did all the paperwork and worked with a few others, and we got the PhD approved. And
When you went back there again, first after your undergrad, after Germany, what did you teach there, or what were you doing?
I was
then?
teaching growth and development and exercise. I was teaching the scientific basis of training. Yeah, a couple of classes like that.
Okay. Okay.
And I was also starting some research, even though I had not done my PhD. I was the person with the most research experience.
Yeah. So, Claude, where then in this chronology did you first hear about the American College of Sports Medicine?
So, when I got back to Canada around 64, after Germany, I was involved with a whole group of Canadians in launching the Canadian Association of Sports Sciences.
I remember.
I was a founding member of that, which is now the Canadian Society of Applied Physiology. So I was a member of that, and I was on the first executive committee. I became president of it, etc. And we had joint meetings with ACSM a couple of times, one in Toronto I can remember in particular. So I went to some of the meeting of ACSM, but I did not join as a member until 1976, when I was in Austin.
Okay.
I joined at that time.
Okay.
And I've been a member ever since, obviously.
Okay.
So that's, I also, since I was training students in genetics, and nobody was talking about genetics but our group at that time, so we had a lot of opportunities to present our work, and became fairly visible quickly because we were the only group. So my doctoral students, the postdocs, we had some sessions at ACSM when all the papers were coming from my lab. That was an interesting period.
Is it safe to say then that genetics and physical activity connection was first presented at ACSM with your students in your own research, or were there other venues out there that you guys were working with too?
There were a few other minor venues. The most important work was presented at ACSM.
Okay.
And this lasted until 1999, when I was offered a position in the U.S. as executive director of the Pennington Biomedical Research Center in Baton Rouge, in Louisiana. So in 1999, I retired from Laval University.
Oh, wow.
They made me professor emeritus of medicine, and I went to BatonRouge, and I've been there for 13 years now.
So, and that's part of LSU?
It is a campus, one of the 10 campuses of the LSU system.
I see.
We're not on the campus of LSU where the Tigers are.
Okay.
We are an independent campus.
Yeah, and it's all the is the medical school there too, or
No,
what?
medical school is in New Orleans.
Okay.
And in Shreveport, we have 2 medical schools.
So do you have students there then, or is it all research, or do you have classes, or how is that set up?
It is a campus entirely devoted to research, no teaching, just postdocs.
Very nice.
And the faculty doing research. About 600 people, and the focus of the research is disease prevention, which was up to my alley.
Perfect.
Yeah.
Are you supported mostly through grants, or is there some state support as well?
About 20% of the funding comes from the state. The rest is all grants and contracts.
Okay.
So it's very competitive. Everybody's on soft money.
Yeah. So then, tell me a little bit about your lab there and your operation. You must have a lot of postdocs and...
Well, I was an administrator for 11 years, so I just decided not to renew my term, and I'm now back in research.
Oh, I didn't know. Okay, I see. Okay.
But now I am heading the Human Genomics Laboratory at the center, and I work closely with Dr. Tuomo Rankinen, also a regular contributor to ACSM, and we both are re-establishing our program in human genetics and health and exercise and obesity and diabetes, because we are interested in all these areas.
Again, prevention.
Always prevention, yeah.
Yeah. Well, did you publish early on in MSSE then as well? Or?
I don't remember when my first paper in MSSC was, but the first paper, the first genetic paper that really originated from the new enterprise that we had launched in Quebec City was published in MSSC in 1984.
Okay.
And it had to do with the fact that we discovered that when we exercise identical twins with a standardized program, all the twins do the same thing. We had 10 pairs. They did a very demanding program for 20 weeks. And we discovered that there were some pairs that increased their maximal oxygen uptake and improved their heart profile dramatically. And both twins, both brothers or both sisters, were the same. If they were high responders,
They were both
high responders.
Yeah.
But we also had the opposite. For example, I remember vividly in that experiment, one pair of twins, two boys, they never missed a session like the others. They were fully compliant. They made zero gain in maximal oxygen uptake.
Both?
Both.
Zero gain.
Zero gain.
Wow.
Within the error measurement. So that first paper was published in MSSC in 1984.
Yeah. Wow.
And we repeated that experiment several times. We confirmed it, and then that was enough to give us the preliminary data to launch the Heritage Family Study, which has been
ongoing.
It continues to go.
Yeah.
We've been at it for since 1992, and several other members of ACSM have been involved, like Jack Wilmore, Art Leon, Jim Skinner, Tuomo Rankinen, and many other investigators across the U.S. and Canada.
I just took a quick tour through the exhibit hall yesterday, and I kept seeing your name on book after book after book. Some quite early on that I didn't know you had done. Was that something that I thought, one, I'm trying to remember now who you did it with. Was it Haskell?
Yes.
It was a large book sort of on fitness, I think.
Physical activity and health.
Yeah, yeah.
We edited that together with Bill Haskell and Steve Blair.
Yes, yes.
Yeah. Second edition, just
yeah,
out.
Yeah,
Yep.
yeah. That was. I'm trying to remember the year. Do you? I don't want to put you on the spot.
Well,
it was pretty early on, actually. I thought for that, wasn't it?
Was it about 7-8 years ago? The first edition.
Okay, I thought it was earlier.
And the second edition earlier this year.
Okay. Have you done any books yet, just on your genetics and physical activity?
Yes.
Research?
Yes. I have written or edited in English or French.
Or German?
No, not German.
Not German.
About 40 books.
40?
Yeah.
Wow, no wonder I can't remember them.
I get a lot of requests, Jack, to probably because I have gained experience in editing.
Yeah.
And I'm fairly good with language, I guess. So that goes back to my training in Latin and Greek and
the humanities.
The humanities. So yeah, this has served me well in this regard. So And currently I am editing with my friend George Bray, who is a former J.B. Wolfe lecturer of ACSM way back. We are the editors of the Handbook of Obesity.
Oh, wow.
4th edition. This is about 100 chapters.
It's huge, yeah.
It's 2 volumes.
Yeah.
So we are now right in the middle of that one. This is a major effort.
A lot of work.
A lot of work.
Now, when you went to back to Louisiana, were you in charge, the head of, the director of the whole Pennington
Yes.
enterprise?
Yeah.
Wow.
Yeah.
That's
We have.
that's a big.
When I left, we had 55 laboratories. When my term ended, we had 55 laboratories.
Wow.
Mostly in basic science, quite a few in clinical, and some in population science. And many distinguished ACSM members are there. Tim Church is one person
Yes.
who was there.
Yeah.
Tuomo Rankinen, Peter Kaczmarczyk, many others. So
Yeah.
we had
Wow.
a nice group of people.
And so that was your administrative stint. Now you're back
Yeah,
doing
research.
your own research.
Really my preferred.
A lot of people
Occupation.
say that
Yeah.
they did the administration and they're ready to move on now.
Yeah, it's not comparable. The challenge, the intellectual joys.
They're not there.
No,
You get all the problems that come to you.
Yeah. but it was a challenge, and I enjoyed doing it. I grew the center enormously while I was there. We almost increased 3 times in size. But it's not the same as the joy of science.
Right, right. What's going on now that in this whole area of genetics and physical activity? What's sort of the latest you guys are doing?
Okay, so this is an excellent question. The tools have changed so much now. You know, when I started my program in the late 70s, I was elated at one point because based on blood cell markers and proteins in the blood, etc., we were able to type approximately 60 markers in people. And I was saying to my students, think about it, we now have 60 markers. And of course we didn't find anything because we were all naive at that time. All over the world we thought that with markers like that we could identify links to
But
disease.
it wasn't enough.
Oh, no. And now, probably in a few hours compared to weeks to type these markers 20, 30 years ago, now in hours we genotype 500,000 to 1 million markers in people.
Wow.
And sequencing the whole genome is within our reach. It is now, the cost is about $10,000. You remember that when the sequence of the genome was published early in this century, around 2001, that enterprise had cost $3 billion. Wow. And today to sequence same
It's
thing,
$10,000.
it's $10,000, and everybody agrees that within a reasonable period of time, maybe a year, 2, 3, it will be below $1,000 to sequence a person completely. The whole 3 billion base pairs of DNA. 3 billion. So this has changed the way we do,
How
we do
exciting.
research.
Yeah.
Completely.
Yeah.
So given this now, we can really use the tools to provide concrete information. And one of the things that I am focusing now on is, I think, the first step in personalized medicine, personalized genomic medicine, with application to exercise. We know that personalized medicine is going to be the wave of the future for disease prevention, right? Focused on what are your weaknesses individually, at the individual level, really. And this will be true for nutrition, for exercise, for smoking, for alcohol consumption, for pharmacotherapy, etc. So in the field of exercise, the first order of business is to identify what are the predictors the propensity to develop adverse responses, misadaptive response to exercise. So that's the first order of business. That's what we want to do. That's what we are now looking funds for at NIH. And if funded, I think we will be able to succeed in having this. Once that first step is accomplished, people could be screened readily with a drop of blood, and we can tell an individual, maybe this exercise for you is not the way to go if you want to lower your blood pressure.
Okay.
You will do it for something else.
It's not, that wouldn't work for that person.
For that person,
I see.
This biological pathway, it may not work for you. There's a high risk that it will not work for you.
Yeah.
So either you have to be very closely monitored or look for alternative therapies, maybe driven by diet, etc. And then we'll go further with more refinement of this, trying to maximize the benefits that a person can experience with exposure to exercise. But that's the next step. The first one is to really trying to identify those who might be hurt a little bit by being active for particular risk factors for heart disease or diabetes.
Okay. So that leads me to the article I was mentioning that one of my students just sent me from the New York Times, that exercise may in fact increase heart risk, right, for some people.
Okay, let me elaborate on that. This is the result of the Heritage family study initially. I looked in Heritage at the range of response for common risk factors. Let's take blood pressure. So I noticed that most people registered a decrease in blood pressure, even though they were normotensive. They were not hypertensive individuals. They were normotensive. But the trend was for a lowering of systolic blood pressure, which is good. Which was
as they exercised.
After the training, after
the
20
training
weeks
bout. Okay.
of exercise.
Okay.
They had a lower blood pressure than before.
Okay.
But there was a subgroup that was not diminishing, tended to increase a little bit, and others who had a real increase. So I said, is this real? And I look at HDL cholesterol, the same. Some people had a decrease in their good cholesterol. Intriguing. And I continued through a number of risk factors, and we were making the same observation. So I said, okay, how can we move this forward? The first order of business, I said, is to identify a reasonable threshold beyond which we can say this is an adverse response. You cannot just eyeball this. You have to have a threshold, a firm threshold. So in Heritage, we had an ancillary study where we had taken 60 people, measured them 3 times over 3 weeks. So I said, okay, here we have human variation in blood pressure is embedded into that, and the error of measurement as well, because we measured the same people 3 times. So we calculated the standard deviation for each individual. And I used this to define the threshold. It had to be beyond that. I'm going to not go into the details, but we use this.
Yeah.
Then, using this, yes, true enough, in Heritage, it was about 8-9% of our subjects who had an adverse response for blood pressure and for HDL cholesterol. And this was true in the whites and in the blacks of Heritage. So I said, okay, we cannot publish this. This is potentially too explosive and risky if we're wrong. So we need other studies. So I went to my friend, Bill Krause, who is also a devoted ACSM member, Tim Church and Steve Blair, they have studies. Jim Hagberg also has studies, and a colleague in Finland, Dr. Hakkinen. So we had access to their data studies that were different, large number of people, some were had more risk factors, others less. We ended up with 1,700 people.
Wow.
And true enough, in all studies we had adverse responders. The prevalence is about 10% for a given risk factors. 10% of people increase their resting blood pressure instead of decreasing it. And we had 7% who experienced this for at least 2 risk factors.
Wow. Out of 6?
Out of 4.
Or 4, okay.
So that's what we published in the journal PLOS ONE, and that's what the New York Times was so interested.
That's what they picked up
Yeah.
on, yeah, okay.
And now it's, of course, it's out of my control.
Yeah. Well, as soon as, because right away people think, oh, gee, exercise isn't good now, And I think one of Haskell's response was people were looking for reasons to not exercise.
Of course.
And now they have one. Maybe I'm one of the 7%, right?
Yeah. And that's a real point, and we discussed this a lot among the authors, because we all believe that exercise is by and large extremely beneficial for people. But we also have to look at reality.
Yeah.
And if this exists, and we think it does, then we need to find the tools to alleviate this, to prevent people, to protect them, to offer alternatives. Maybe some other types of exercise might be better for them. There's a whole field of research that now needs to be mined.
But what you're saying, though, is that with your genetic sampling, you should be able to determine who these people are.
Even before they start exercising.
Right?
Yes.
Yeah, okay.
So that's the goal.
Sure. That's the neat thing, because then you can identify those that would have a propensity for
Absolutely.
the blood pressure going the wrong way.
And it's true. It's true in prevention, but it's also true in rehabilitation. For example, cardiac rehabilitation programs.
Yeah.
These people are there. And,
True,
and the cardiologists tell his patient, you need to go to this exercise program.
And it could be doing them harm.
And it may increase their blood pressure.
Yeah.
And decrease their good cholesterol, increase their triglycerides, etc. So that's good there. It's good for also for applications in the prevention or control of type 2 diabetes. I can see a whole variety of areas where this will have potential for helping participants.
Absolutely. But before you can do it, the $10,000 has to be reduced, I guess, to make it practical.
Well,
somehow.
I think it could be. It is independent of that, because if we are successful in getting this grant and identifying the genomic predictors, it will be a subset.
Oh, just need you'll just go look for that.
We could devise
Sure.
An array that could be used to genotype only these
rather than the whole thing.
markers. Yeah, maybe 50, 100 would be a simple thing.
Okay. I'm very naive about this whole field, so forgive me. But so are there then markers that you've identified that just relate to obesity and physical activity, or different markers you've identified, I guess is what I'm asking.
We have, particularly for the response of maximal oxygen uptake to training. We focused on that because we can measure maximal oxygen uptake quite well, and and we use it as an indicator of fitness for athletes, for participants in health programs, public health programs, etc. So we focused on that, and we have identified a panel of genetic markers that even before people begin exercising, we can use to predict how good they are going to be in their response.
Okay.
And it works pretty well.
All right. Okay. So a coach could have an athlete, a prospective athlete, that you could identify, and they're going to, well, all the training in the world isn't going to make this person a distance runner.
Yeah, if you are not a good responder, the probability that you are a material for world-class endurance performance is low.
It's not going to happen.
Is low. Very
Not
low.
likely to happen.
Not
right?
likely to happen.
Okay. Gee, we're almost out of time. So you're doing a session here?
Yes,
Is it this
exactly.
year?
Exactly. I'm speaking in a session that will, it's a kind of a debate on the role of training versus nature versus your endowment in the ability to become a world-class athlete.
Okay.
So that's not really the area I work on, but the fact that we do genetic research offers a platform for bringing some light on this debate.
Oh, absolutely, yeah. That'll be attended well, I'm sure, today. Well, thank you, Claude, very much. I enjoyed talking with you, and I always watch at MSSC. It seems like every year you have that update.
Yes.
Keep of the new stuff.
That's right.
you know.
Every year we publish, a group of us, all ACSM members,
Yeah.
we publish an update on the genomic science.
Right.
What have we learned in exercise genomics in the last 12 months?
Yes, that's very neat.
It's been very rewarding. It's very well received. It's one of the most cited, I think it's the most cited paper in the whole of MSSC.
I bet it is. And what is neat is that you were onto this so early.
Yeah.
You know, that's really very, you were doing the right thing at the right time.
That's among some of the things I did right when I was young. There are a few other things that we will not talk about
We won't
here.
talk about today, right? Well, thanks again, Claude.
Jack,
I
that was great.
appreciate
Thank
it
you.
very much. Thanks again.
Thanks for the opportunity.
Thank you.